Tonight's presentation is it's an update on prostate cancer research, and a lot of the research now that people aren't quite aware of is being funded through some incredible fundraising efforts through Movember. Tonight we've got Dr. Morley Hollenberg and Dr. Hyndman. They were recent recipients of a Movember research grant. It should be noted that last year, Canada alone raised over $40 million in the Movember campaign, which from really a social media aspect is truly phenomenal. The majority of the funds go directly to prostate cancer research. Last year there were 40 individual grants and rather sizable grants that were awarded for various aspects of prostate cancer research, and they focused largely on basic research, advanced disease, survivorship and quality of life issues, and biomarkers and earlier detection. We were also happy to earlier this year announce that there was a $5 million team grant awarded. The primary investigator for that was Dr. Ralph Butchin out of Vancouver, and it was for treatment of, granted for the research of treatment-resistant prostate cancer. We also gave away 4 Rising Star Awards for new prostate cancer researchers. Dr. Hollenberg and Dr. Hyndman's research is entitled Kallikrein PSA Signaling Proteinase Activated Receptors, or PARs, and Prostate Cancer Development. Dr. Hyndman is a second-generation urologist here in Calgary, and he's really making a name for himself on the research end of things, as well as the nerve-sparing surgeries that he's doing in the urology offices. Grown largely for most of his life here in Calgary, he did do a fellowship program at Johns Hopkins University, and I'm going to let Dr. Hollenberg talk a little bit more about him after this talk. But I would also like to point out Dr. Hollenberg's accomplishments, because they're quite notable. Since the late 60s, Dr. Hollenberg has made significant contributions in the areas of molecular pharmacology of hormone action as related to signaling by hormone-like mediators. His scientific contributions have been recognized by the membership in the Royal Society of Canada and a number of research-related awards, including the Pharmacological Society of Canada's 1999 Novartis Award for Outstanding Contributions to Pharmacology in Canada, the Canadian Society of Clinical Investigation's 2009 Distinguished Scientist Award, the Royal Society of Canada's 2011 McLaughlin Medal for Sustained Research Excellence, and 2 of the more recent awards are the CSCI Royal College Henry Friesen Award in 2012 for demonstrated leadership in developing biomedical research at local, national, and international levels. He was also awarded this year the E.K. Fry eWorld Commemorative Gold Medal for outstanding contributions on subjects in health and disease and with significant influence on the development in these areas. Dr. Hollenberg was born in Winnipeg and is really seen as into a family that's known as could be considered a Canadian medical dynasty. His parents were physicians, 4 uncles, an aunt, and many cousins are also MDs. Morley's first pursued his training in chemistry and pharmacology and not medicine. Having obtained both undergrad and graduate degrees in chemistry from the University of Manitoba, he went on as a Rhodes Scholar to obtain his PhD in pharmacology at Oxford in England. At that point, his interest in medicine took over, and he attended the Johns Hopkins University School of Medicine, where he received his MD degree and clinical training as a medical intern at the Johns Hopkins Hospital. From there, he obtained postdoctoral training appointed as a member of the Hopkins Departments of Pharmacology and Therapeutics as a Howard Hughes investigator. Moving then in 1979 to where we were fortunate to bring him here to the University of Calgary. He's headed the Department of Pharmacology and Therapeutics for over 10 years and continues to serve jointly as a member of both the Department of Physiology and Pharmacology and Department of Medicine. His laboratory has thrived over the years with a focus on hormone-like roles of proteinases and the earlier mentioned proteinase-activated receptors or PARs. To this day, he participates vigorously in the research and teaching activities at the University of Calgary. He's also added extra roles in his spare time as a co-director of the faculty's Leaders in Medicine program, which trains MD PhDs and MD master's students who will create the medicine of the future. Now, on a personal side, he's also a professional artist whose focus is on Chinese brushwork. And in fact, his work can be found in international collections. On a family level, he's got a very supportive family comprising of his longtime partner, Joan, 2 children, Elisa and Daniel. And this has grown to include his children's partners and 2 grandchildren, Myra and Isaac. Thus, issues of science, art, and family frame the essence of Morley's future. Please join me in welcoming tonight's honored guest and the recipient of the Movember Researcher Grant Award, Dr. Morley Hollenberg.
Well, thank you, Steve, for a very generous introduction. I really appreciate that a lot. And it's a privilege to be here amongst you because, as I'll point out, I work for you, not the other way around. You pay my salary at the university, and thanks to you, the lights are on in my lab because people have helped a lot with the Movember grants. And I want to tell you what we're trying to do with Eric and myself. And the key here is that this is a collaborative atmosphere that we work in. We build teams. And you're part of the team. You got to know that you're part of the team because you are as much a process for the discovery of what we want to discover as Eric and I are and the people that work in our lab. And so what I've listed here are just a number of names, but in particular Eric and the student between us, Olyemi Felligan, working on some of the NMR and some of the culture stuff. Brian Donnelly, who, as you know, is heavily involved in our Prostate Cancer Center. Tarek Bismar is one of the pathologists. Kirill Tripkov, another one. And these are all individuals that have come together as a team to solve a key issue that we're dealing with. And we've collected a collaborator in Finland who actually has taught us some of the culture methods that I'll talk about, and who is integrating his work with ours here. So in Finland, he's looking at some of the genes that are produced by cancer-producing prostate cells, and he's collaborating with us, and we share emails across the ocean and phone calls. John Lewis up at University of Alberta is building, we hope, a real center of prostate cancer research in the province. I have a longtime collaborator, Eleftherios Diamandis, who's the king of PSA. He absolutely knows more about PSA than anybody almost in the world, and Keith Jarvie, who's a urologist working with us. And so we spread across the country and we're across the ocean building a team to work on the problems that I'll talk about. I belong to this Inflammation Research Network. It's a group of us at the faculty. And cancer is an inflammatory disease. In other words, cancer triggers all the hallmark signs of inflammation: increased blood flow, pain, swelling, fibrosis, and all that kind of thing. So I work in this inflammation network. So we consider cancer as part of our bailiwick. And what I'd like to talk about today is about prostate cancer behavior. And I know that all of you in this room will be familiar with the abbreviation of PSA, and I always ask people, and you'd be surprised how many people don't know what it is, but when you tell them what it is, they say, oh, it's some kind of a test for cancer. Okay, prostate cancer, all right. And what most people don't know, and I'll show you where I learned it from first, is that this is an enzyme. And an enzyme is a long string of amino acids, a protein that has, and this particular one the PSA family does, is it chops up other proteins. So that's the meaning of the word protease. And there are lots of highfalutin words in the title, but I'll try to translate them in a way that you can go away and if you didn't understand them to begin with, understand. Because everybody knows that you digest your food. And most people know that there are enzymes that do the job for you in your stomach. And PSA is that kind of an enzyme. It digests things. Not your food, but it digests other proteins. So I'm going to talk about the PSA family because it has 15 brothers and sisters in your gene. And I'm going to talk about how they tell cells that to wake up and do something. So they signal to cells. It's like somebody coming to the door, putting their finger on the doorbell, and the doorbell rings. So what the PSA enzymes do is they click that doorbell, and I'll show you how it clicks. And they tell the cell inside, somebody's outside here, you better start doing stuff. And they tell the cell very specific things to do. And by knowing how those signals work in the cells, Eric and I and our colleagues are hoping to figure out a way to track the progression of cancer. Because the challenge is this. You know better than most people that I have prostate cancer. Because anybody who gets to my age, when you take apart my prostate, you're going to find some cancer cells there. But the difference between me and many other men is that, hopefully, my cancer is asleep at the minute, and it's not awakened. If you look at it, it says cancer, but it ain't doing nothing there. So that's the indolent or quiet one. And what we need to do is to figure out how to look those cancers in the face and distinguish the aggressive ones versus the ones that are going to sit there and outlive you. So that's the job we have to do. That's the challenge we have. And as you know, to meet that challenge as of today, we still are not very far along. We have pretty good tests. They help us a lot. But as all of you know, the PSA test, which is a good index of whether the prostate is producing that enzyme or not, is very good to follow cancer. If you had it and had your prostate out, it's really good for that because it'll tell you if the prostate cancers come back. It's sort of okay to kind of follow what's going on in your body, but if the test is taken out of context, it can make you either very upset, like my dad who came home one day and he says, my PSA is up, you know, I think I've got prostate cancer. Well, he had inflammation of the prostate. That's what it was. He didn't have prostate cancer. He died of something else. Or people that follow it and your prostate PSA goes like this, boop, and then you have to have your prostate biopsied and take a look at it. So that's one test that is a little bit controversial. And then there's the tried and true Gleason score. So a biopsy's done and the pathologist looks at it and says, that's really good news because it's Gleason, very low number. Or you know what, this is not good news, the Gleason's a very high number. And so that's looking at the pattern of the cells in your prostate. And the pathologists know when they're looking those cancers in the face if it's obvious or not. But if it's like the cancer that's in me at the minute, they couldn't tell that it's going to change or not with time. And so I've had my biopsy done, and it was pinned, whatever, and I'm kind of following it, but I have no idea what it's doing. I'm hoping it's quiet. So the challenge we have is how are we going to follow on with these tests, which are okay, but we really need to do better. And so I'm not sure if this is readable from the back of the room, but I'll read it out. So what Eric and I are trying to do is to use what I call the fourth dimension. So a number of you are in the active surveillance program. That's the fourth dimension of prostate cancer. My dad always said, he was a physician surgeon, he says, time will declare the diagnosis. You got to follow it and follow the track. So what we're trying to do is use this fourth dimension and capture the changes that happen when a cell goes from a non-invasive cell to invasive cell. And if we can capture the right picture that characterizes invasive versus non-invasive in biochemical terms, we're hoping that we can take a look in your prostate when in the active surveillance say, you don't have to worry just yet, or you know what, things are not looking so great, we better do something more. And so what we'd want to do is to characterize, and I'll show you the kind of cultures we're using, with the pathology that we can see under the microscope with our pathology collaborators to match the picture between what we see in our model system and what's going on in the prostate. And by following individuals with the active surveillance program, we're aiming to be able to follow the fourth dimension of what's going on. The other thing is to follow the change in the picture at timed intervals during the active surveillance in people. So what we're doing is we're doing active surveillance in a culture system, which is cells grown in a test tube in my laboratory. Well, what good is that to you? The answer is we don't know, but we must follow you. That's where you come into the picture to help us follow as we keep surveillance going in people who are at risk. And what we'd really like to do, along with Dr. Diamandis and company in Toronto and Eric and the NMR on campus, is to develop some new biomarkers so that we may not even have to biopsy you, but we would just take a sample of all things semen, which we think will carry some of the messages out of the prostate into the external world that we could analyze and say, you know what, it's reporting on something that we can correlate with cancer that's going to stick around for a while or cancer that's going to get up and move too quickly. So the idea with our work is that we will do the fourth dimension business, characterize what's going on in a culture, match it with what happens in the tissue, and then, with hope, identify a marker in the semen, which Dr. Diamandis says is the biggest wasted body product in the world, or something like that. There's your humor for the day, anyway. And try to get some sense out of that. So how does this PSA family, how do they talk, how do they communicate with the cells? It wasn't known that they communicate until I came across some information. I'll show you how it came about. So here's a common mentor between me and Eric, Donald S. Coffey. Now here's a guy who is a biochemist who woke up one morning at Westinghouse with a good job and says, I got the call to cure cancer. And so he quits his job, a couple of kids, goes to Hopkins, gets a PhD in Leslie Hellerman's lab, and becomes the world expert in prostate cancer and has generated people like myself and Eric who are absolutely committed to the disease. No MD whatsoever, but he led the Brady Labs in research, and I'll let maybe Eric do some anecdotes if he can. So Donald Coffey was one of my teachers in medical school, and he was working with some of his students, and he says, by the way, and they were looking in dog semen because the dog is the only animal model of benign prostatic hypertrophy, and he says, you know, we've discovered that this major enzyme in the dog semen is a proteolytic enzyme, is a protein degrading enzyme of a certain kind, and he published with Bill Isaacs and company back then after he'd talked about it in our medical class. And it was that piece of information that kind of stuck. That, okay, so PSA, most docs don't know, is a digestive enzyme. So you're already far ahead of, I'd say about 85% of most medics in our medical school anyway. So here's what a digestive enzyme does. So your food goes in, and it's made up, so each one of these letters stands for an individual building block in proteins that you eat. And so here is the protein sequences. And what the enzyme does is it chops between these building blocks at certain places that the proteins, that the enzymes like to chew on. And so once the protein has been acted on, you've got these pieces that are then floating around. And those pieces could be coming from, this might have the sequence of insulin buried in it. And so when the enzyme chops, it reveals insulin that gets into your bloodstream as insulin, whereas it was pro-insulin before then. And these cleavage products of digestion can be active, and they can stimulate prostate cell function. So that's the mechanism that the PSA family uses to communicate to the environment and the prostate cells. It chops up proteins in the environment, and those chopped products can signal to cells in their environment. So you can create hormones like insulin by this method. You can spring sectors. It's like they're attached on a spring, and the enzyme comes in and cuts the spring and unsnaps it. So there are ways that the enzyme can do that. It does it to the insulin receptor in particular, without insulin even being present. It can digest the material on which the cells are sitting. So there was a lot of excitement because there are enzymes that lift cancer cells off, or enzymes that the cancer cells use to get through the cell membrane. And they thought, well, if we block those enzymes, we can cure cancer. And they cured a lot of cancer, except they only cured it in mice and not people. So we got to do better. And then there is the mechanism that I'll talk about where the enzyme tickles the cell surface, gives it a signal, and then the signal gets translated inside the cell to say, aha, there's something out here, and we're going to ring the doorbell. So this is these are the digestive enzymes in the center of the story, and they can do all these things. They can activate a receptor, they can generate these peptides, but we're going to talk about these proteolytically activated receptors. That means that a digestive enzyme, like the clotting enzyme in your bloodstream, when you cut yourself, two things happen right away. You start to bleed and then it clots. That's a digestive enzyme in your bloodstream called thrombin. And when thrombin causes the blood to clot, it also scratches one of the cells nearby to say, this person's been injured, you better start growing, or here's some pain for you, you've cut yourself over here. So I'm going to talk a little bit about the way these digestive enzymes signal to cells. And the acronym PAR, proteolytically activated receptor, has been aging for about 22 years now. Some of you may or may not. And so back in 1991, the first of these receptors was cloned in both sides of the ocean, and I got interested in that. And shortly thereafter, the human receptors got cloned. And they act in the following way. So here is the digestive enzyme up here, right? And it chops a piece of the nose of the receptor off. So you leave behind eceptor, and this other part goes away. So this eceptor part is unmasked, and it swings around and gloms onto the outside of the cell and kicks it in the backside and starts to signal. So that's the way these receptors work, that they get cleaved on the outside of the cell, they reveal a piece, and I'll show you again, and then they swing around still on the outside of the cell, and they transmit a signal to the inside of the cell that causes the cell to do things like grow, invade, or die. So it just depends on how the cell receives the signal. And here's my rendition of the receptor. So it kind of sits in the membrane, winding its way through the fat part. And there's an end here that gets chopped off by the proteolytic enzyme, the digestive enzyme. And it reveals this little part here that's like this little red dot in the middle of the circle here. And then it can swing over, or what we can do is we can synthesize something that looks just like this, like just a little part of it, and it can fool the receptor into activating as well. So we have this tool that we can kick the receptor on the cells, and the cell will do what it's going to do if the PSA enzyme were there. So that's key, because we can study what the signal would do in the cell even if the enzyme's not there. It's like having somebody else ring the doorbell for you, if you look at it. And this is the way the biochemists like to. So this receptor sits in the cell membrane, and it's got this outside part that gets cleaved, and we can play around with that by blocking it with an antibody, and it sends the signal inside the cell by talking to so the outside talks to the inside, and the signal goes into the cell. And if you have this little piece of the receptor up here, the slide rule it says, then it can activate the receptor too. So that's how the receptor works for our system. Now, how did I get to where Eric and I are today? Well, it starts here. Now, on your left is Charles Huggins. Now, Charles Huggins got the Nobel Prize because he figured out that prostate cancer was dependent on the androgens. And a lot of you know that. And the big team grant was given to try and work with prostate cancer that is not dependent on androgens. So Charles Huggins was a mentor for my mentor, Paul Talley, who, along with Don Coffey, were involved with my education at Hopkins. And Paul took his cue from Charles Huggins, and he says, I was inspired by the revelation that even advanced and disseminated cancer could be controlled. And from that time on, my research interest never strayed far from the cancer problem. So it was Don and Paul who actually put the hook in me to get me to the place where I'm standing up before you tonight. And it's that kind of inspiration that I come from. So what about this connection between the PSA and the abbreviation for the fancy family is callicrines? I won't go into the derivation of that Greek word which means sweet, etc. But what is the connection between those enzymes and these proteolytically activated receptors? Well, I knew from my medical school days that the prostate made the enzymes that could activate the receptor. Then, and Don had told me, that this is a serine protease, and we knew that these enzymes were responsible for knocking the nose off the receptor. So putting it together with these family of 15 digestive enzymes that are widely expressed, okay. So then it turns out that there are 15, including PSA, there are 14 other members of the family. And they're all over the place in the body. Of course, what we know in this room is that a lot of the PSA, so that's number 3 out of 15. And it is in the prostate, but it's found in other places in the body. And you're not meant to read these letters, but just to say there are a bunch of these family members in a bunch of different places in the body. Not just the prostate, so they could drive cancer in many places. So when we crack the code of prostate cancer, there's a chance we'll crack the code of a lot of other cancers in which these enzymes are playing a role. So the PAR family, we know, is activated by these digestive enzymes, and it's also widely distributed and expressed in the prostate. And what these black dots on the screen mean is that every time you analyze the tissue and do a special test and run it on this gel, if there's a black dot, the enzyme is there. If there's no dot, then the enzyme is absent. And what I realized in this first paper that came out characterizing where the receptors were was that there are lots of black dot that's in the prostate. So here we go. The prostate has the enzyme that can activate the receptor that's also in the prostate. And I thought, well, maybe they get together. And that was the cue for me to get on to Dr. Diamandis in Toronto, because he was the expert in these enzymes. And sometime after that fact, Eric and I got together with Matthias Nies, who's in Finland, and what he did was he went into the database that's at the Sloan Kettering that has all the prostate cancer cells and all the kind of tags, the molecular tags that are put there. And we said, cancer, do you have one of our favorite receptors, PAR number 1, and it's got a fancy name now, F1, F2R, etc., blah, blah. And they were able to look at the survival curves. So what you're looking at here is that if you've got low PAR number 1 in your prostate, your survival curve is above that for high PAR. Now, you don't do better in the sense that you live forever, but it certainly is an index that this receptor of this family has got to be involved somehow in driving cancer in the body. So we have circumstantial evidence that these receptors are involved for survival, and we know if you look in the tissues that these receptors go up, down, and sideways in all kinds of cancers, including cancers of the prostate. So not only does the PSA go up in its family members, but the receptor goes up too. So we developed this model. The working hypothesis is, can you imagine a way in which this process would work in the prostate? So we have this working hypothesis that the family enzymes would signal and talk to these receptors. And we got together with Dr. Diamandis and a very gifted PhD student, Katerina Okonomopoulou, who took it on herself and was impassioned by trying to put together these KLK enzymes. So these are all members of the prostate-specific antigen family, number 14, number 6, number 5. And what this test is doing is asking whether the enzyme on the outside of the cell can send a signal to the inside of the cell. So the signal that we're looking here is like a light goes on inside the cell. And literally the way we look at it, the light does go on And then when the light goes on, it says that calcium has gone up inside the cell. So this peak here is like light going on in the cell, and then the light dims. And if you put in KLK number 14 or KLK number 6 or KLK number 5, the light goes on, and that says immediately that the KLK family, the PSA family, can talk to cells and turn the lights on inside the cell. So if we do that with a biochemical test that puts the light on, can we actually see? So I wanted to bring you a picture that shows you how the cell is actually looking and responding to the enzymes. I don't know if this can be seen without the lights dimmed, at least over the screen. Does anybody know how to work the lights? So what has been done here, if you look on the bottom, is that we've put a green tag on the tail end of the receptor that sits inside the cell. I think, can you see that from the back? Can you see the, yeah, okay. So we put a green tag on the back end of the receptor, and it's actually sitting inside the cell. And this other part that gets cleaved is outside the cell. So when it's sitting on the outside of the cell in this version, the green tag is ring around the rosy. It's all around the outside of the cell. It's just sitting there. But when PSA KLK number 14 visits the cell, what you can see is all the dots kind of cluster and get inside the cell. So they're going in, you're actually seeing the footprint of a receptor activating a cell. And you can actually watch this sort of in real time. This is just a scientific way of quantifying how many dots go in, and just as a lot of dots go in, whether you activate with a trypsin-like enzyme or one of the PSA family enzymes. Now, two people in my lab, Koichiro Mahara, who is being supported by the grant that we've just got, and Rithvik Ramachandran, developed a really cool way to look at these receptors before and after they're activated. So this is par number 1, and it's got a red tag on the outside and a green tag on the inside. So when they're together, you see yellow. So before you put in the enzyme, the yellow's all around the outside of the cell. Now I told you about thrombin, the coagulating peptide protein. When it sees thrombin, the cell goes nuts, and all those little green bodies go inside. Or in addition, I've told you we can make a piece of the receptor that looks like this SFLLR piece, and it too drives the cell. But what we know is that it hasn't cleaved the cell, it's driven it inside because all the dots in there are mostly yellow. So you're seeing a visual activation of a cell by a PSA-like enzyme on the outside of the cell, ringing the doorbell and lighting up the inside of the cell. So what happens in a prostate cancer cell that might be, that was kind of a normal cell, it was an embryonic cell, a human embryonic kidney cell, that we're able to stuff these receptors in and get them there. So this is a prostate cancer derived cell line, so-called PC3, that either does not or does express an androgen receptor. So we can pretend that the cell is either androgen resistant because this cell on the left does not respond to androgen, or it does respond to androgen, although we're not feeding it androgen in the system. We can tell what happens. However, there's a big difference when we look at this cell for two reasons. First of all, I don't know what the heck all these yellow dots are doing in there, but those are unactivated receptors that are sitting inside the cell. And if they're on the outside of the cell, they should look like this. However, what this says is that they're all green and sitting outside the cell, and what that means is that there's an enzyme that the PC3 cell has produced, and we know it makes kallikrein number 6, and it is stripping the receptor on the outside of the cell, but it's giving a different signal. So it's not ringing the doorbell inside, it's ringing the doorbell just at the door. And when androgen receptor is present, the cell looks even different. So you've got all these green dots, which by definition are activated receptor, and they're both on the surface and inside. So this is one of the first looks we've had since we started the project as to what does the receptor look like on a real prostate cancer-derived cell, and the answer is the cell may be talking to itself a lot and actually activating itself by itself without any other cues. Now, what this picture shows when it's dark, is the number 1 receptor, the proteolytically activated receptor 1, and this is out of the work of Dr. Barshavat in Israel, who's done an amazing job to show how these receptors drive invasion. And what she's saying is, you know what? When we've got a tissue from a person before they've had their androgen withdrawn by androgen ablation therapy, there's lots of this receptor, chock-a-block in the black. But when the androgen has been removed by either an androgen antagonist, so-called, then all the black goes away. So that right away tells us that not only are these receptors there, but they can go up and down depending on how much androgen is there. And actually, if one looks really closely, although there's a lot less receptor, there still is receptor present. So this kind of fits together a little bit with that survival curve I showed you. If you've got lots of this guy or gal around, you don't do as well as if you've got not very much of it around in terms of survival. So that is putting together the circumstantial evidence that in a tumor tissue, you can have the receptor go up and down. And what this picture shows is a colon cancer, collaborating with Dr. Darmoul in France. And this piece of tissue is very near where the colon cancer is. And these 3 panels here, which have a very different color than blue, they're not blue because they're brown in reaction with an adenocarcinoma that is making lots of the kallikrein number 14, another brother of the PSA enzyme, and it's chock-a-block in this tumor, and we know the tumor also has the receptor. So here again, the principle is that these enzymes can regulate signaling, that they're made both by the tumor and we know by the normal tissue neighbors. They're increased in the setting of prostate cancer, and high PAR 1 is poor outcome. And therefore, these enzymes and their target represent a target for therapeutics. And so I'm going to show you how we're trying to capture this 4th dimension in the culture so that we can get a handle on how this is going. So the 4th dimension is to document the changes in the enzymes and their signaling via these receptors and try to get a snapshot. And then we'll try to match that snapshot with what we can get in the tissue biopsy with the active surveillance program. Now this is a picture of a breast tumor-derived cell. Kari Shemenko is a colleague of ours on campus, and we're doing these cultures in 3 dimensions. And when the cells go there, they kind of look packed together and they kind of stick together. But over a period of 2 weeks, these cells transition from non-invasive to invasive. So it doesn't take much imagination to know that these arms are sticking out into the glue and they're becoming invasive. And what we want to do is to capture a biochemical snapshot of the cell here and compare them and find out what goes up, down, or sideways when the cell goes from a small ball to an invasive ball. And working with us, between Eric and myself, Olyemi has started to work with some of the 3D cultures, and she's looking at the PC3 that I showed you is clipping its own outside, and so the PC3 cell goes from small balls to pretty big balls at day 24. I haven't got a slide to show you all the fingers in here. And when you got the androgen receptor, it looks like it's doing it even more. So again, we're starting to get on the track of the picture of a cell that's not invasive at day 8 compared to the picture of the cell at day 24, 2 weeks later or so, a week or 2 later, that is invasive. And if we can capture the characteristics of the invasive cell and find out what's different from the non-invasive cell, maybe we'll have a handle to look in the tissues. Now, it's not only the cancer cells that are talking. And this is just put up because this is our collaborator, Matthias Nies, and the green cells are actually cells that are not cancer cells. They're cells that will be in the environment for many cancers, the non-cancer kind of the neighboring cells, the host cells, the nest cells, if you wish. And there's a big deal now in terms of how the quote microenvironment, that means the small environment around the cancer, talks to the cancer. And so we should also be able to study how these environment cells talk to the cancer cells. This is just to show that you can distinguish them, and they can be pretty close and talk to each other. And we think they're talking to each other with these enzymes and receptors. So the principles for tracking these KLKs, I'll call them the PSA-like enzymes, is to monitor the transition and take snapshots, measure the changes in the enzymes, measure the changes in the receptor, and match the changes with the cell behavior and catch that snapshot of the non-aggressive versus the aggressive tumor. So here we go, and I've just shown you. So here's the non-aggressive one, looks like that. Here's the aggressive one, and it's invasive. And we'd like to capture, we predict that there'll be low and not very many enzymes, and low receptor, and low proteolytic activity of other kinds. So this is our projected result that we expect to find. And in the invasive ones, we think they're going to have high enzymes, high receptor, et cetera, et cetera. We don't know if that's going to be the case, but that's our working hypothesis. So to test the hypothesis, we grow the cells, we expect these outcomes. If we get them, then we say we're on the right track. So then we want to match the prostate biopsy tissue signal snapshots with the biochemical signal that we see in these 3D cultures. And we'll use these prostate cancer cell lines, but here's where you come into the picture, at least the men in the audience, that we will hopefully take some of the cells out of the biopsy, put them into these cultures, and watch how they change or don't change, and get their characteristics as we go. So we'll both look at the prostate biopsy-derived organ cultures and the model systems that have been derived from cancers, but we know for sure that the model cells really don't reflect what goes on in the body. We just use them as models to test the ideas that we have. Then we'll measure the changes in this signaling profile for the biopsies obtained during this active surveillance and during follow-up. And we'll match the snapshot, we hope, that will distinguish the invasive from the non-invasive from those found in semen. And that's where Dr. Diamandis and company come into the picture, because if we can define with the metabolites or the protein, so here's where Eric is in with the big magnets on campus, that maybe we'll have some, so it's called, fancy name, metabolomics, which just means the metabolic products that are swimming around in a cancer or not in a cancer. And with Dr. Vogel, who's our NMR expert working on the metabolomics along with Eric, we hope to get some of those markers. So that's where we're headed. Now I'll give you a very complicated overview slide to try and sum all this up. So the overview is the cell signaling snapshots to identify these non-invasive versus aggressive prostate cancer. This looks very complicated, but I'll walk you through it. It's not that complicated when I walk you through it. So we've got these 3D cultures, and we're going to take snapshots of the non-invasive, let's call them blue, and the invasive ones, and we measure their characteristics. Then what we do is we look in the prostate tissue biopsy. If we can match the blue signals with the tissue biopsy, then we say, okay, you continue in active surveillance. But if we get a lot of these red signals, then we say, I think maybe we better have you see Eric and get going. And so the key there, though, is that maybe we can replace the biopsy analysis simply by measuring the metabolites and the protein in your semen. And so with Dr. Diamandis, what we'd hope to do with the active surveillance is look for good biomarkers. So you just have to come and donate a sample, and that would be far less risk than going to a biopsy. And based on the sample, hopefully we could make a match of whether your cancer is progressing or not. So that's the picture. It was a little bit complicated, but I felt that I owed it to you to explain in terms that I hope you'll get at least a little bit of how these enzymes that are represented by PSA talk to cells, and how the talking to the cells may have an impact on their behavior as cancer cells or not, and how when the cells change with time, that we could monitor their changes and match the change going from quiescent to aggressive, and match those with what's coming out, hopefully in the semen, and then be able to do the active surveillance in a much more informed way. So that's the nutshell of the talk, and I've got the collaborators up here. Hans Vogel is the one who runs the magnet. I've talked about Nahar and Rithwick, who developed these neat ways of looking at the receptor. Tarek and Carol are the pathologists with us, and Oyemi is the graduate student between us. John Lewis will communicate with at U of A, and Matthias Nies is the one who's helping us with it's called informatics, which just means have your computer tell me what's going on here, because you do the information analysis of a lot of stuff. So that's the talk. I thank you for your patience. And now, I don't know what we should do, have a stand-up, sit-down to get everybody who's fallen asleep back awake again, because Eric and I work as a team, and as I mentioned, we have in common a similar mentor at Hopkins. Eric is responsible for the metabolic consequences of cancer and how the picture of metabolic activity could reflect what's going on in the cancer. So with that, Eric, I'll turn it over to you. I'm not sure how we get to your slide. So I'm not sure how much time I took. I didn't count the thing, but I assume there'd be a trap door open up if it was too long. But,
all right, so now you know why we're so happy to have Morley on our team here. He's truly is I said, can you hear me now? Yeah. All right, there we go. I'll get a little closer to the mic. I was saying, now you understand why we're so happy to have Morley on our team here. He truly is a giant at U of C and around the world in terms of science and cell signaling. So I'm Eric Hyndman, one of the urologists here at the Cancer Center, and I gave you a talk about a year and a half ago on robotics, and I still do robotic prostatectomies. However, another interest that has developed in mine is active surveillance, and some of you may have been involved in that program. So I'll talk a little bit about that tonight, and then move on to a difficult word to say, which is metabolomics, and try and tie those two things in. So the natural history of prostate cancer, as Morley mentioned, is somewhat difficult to predict. If you look at autopsy studies of people who have actually died in car crashes, certainly not anywhere near prostate cancer at the age of 40, there's a relatively significant proportion of them that have some form of prostate cancer within their prostate that probably was going to be dormant for most of their life. Now, if we were to take out all the prostates of all the 80-year-olds in Calgary here, we'd go to jail, but also if we looked at the percentage of patients who actually have prostate cancer in 80-year-olds, it's about 80%. So in my office, I just have an easy way to remember that. You're 80, you got about 80% chance of having prostate cancer. Now, 80% of those people are not going to die from prostate cancer. So there's a huge population that has it or harbors it that is not going to have problems. But there's also a reasonable chance, 3.6% in the red there, that you actually are going to die of prostate cancer. So a very big number has it, and because that's so big, even though there's a lot less percentage of people who are going to die for that, that actually happens to be a relatively significant number as well. So if you look at people who are dying from prostate cancer at the bottom of the slide there, and I'll move along, in Alberta, so these are the cancer statistics from 2012, about 790 men will be diagnosed and die from lung cancer. And if you're diagnosed, you're much more, unfortunately, likely to die from lung cancer, 390 from colorectal cancer, and then 370 will die from prostate cancer, which represents only 14%. So you can see that most people who are diagnosed actually don't die from prostate cancer. So there's a very high diagnostic rate, but again, the prevalence is so high, therefore it still is a very important. All right, so this, you can't, it doesn't show very well at all. These are plots of essentially patients who are the darker are dying from prostate cancer stratified by age. Okay, so down here it's a relatively young age, and then it's also by different categories, Gleason score. So the darker the lines, and as you go down, the more aggressive the prostate cancer is here, okay? And then along the other axis is time. So in one box, you've got, in this group, it's an older gentleman with high-grade disease. They're going to have a high chance of dying as time progresses. Okay? So up top, when we've got Gleason 6 disease, a very small, actually, percentage of patients, and you can't see the actual number, but there's another dark line right here, represents 10% of people or 15% of people are going to die from prostate cancer with Gleason disease at 20 years. So you've got this Gleason score, low-grade disease of Gleason score, where it's going to take an awful long time to cause problems. Whereas if you have a high Gleason score, then generally things develop a little bit more quickly. So you have to keep in mind when you're planning treatment for patients, that's a very important thing, is how long they're going to live, how aggressive their disease or how aggressive you think it is. Now, active surveillance has come to the forefront over the past probably 10 or 15 years, where maybe we shouldn't be operating on these patients with lower grade Gleason scores, especially if they're a little bit older, and they might actually benefit from just following things along and seeing what happens. So this slide summarizes a bunch of the different active surveillance cohorts that are around the world. And I think the most important things you need to look at here is the overall survival and then cancer-specific survival. These are in percentages. All right, so you're doing okay if you essentially have 100% cancer-specific survival. So what this slide is suggesting, and the data from all these different groups around the world, is that active surveillance for low-grade disease works very well, and with cancer-specific survival essentially approaching 100%. So we started a active surveillance program about a year and a half ago here in Calgary, and there are a bunch of criteria. Certainly patient preference or medical comorbidities is a very important feature, and you'd have to figure out how, although it's very difficult, you have to try and figure out how long, or with other medical comorbidities, if there are going to be problems, then maybe operating on a patient even though they're younger may not be beneficial. And in younger patients, again, you'd have more stringent criteria where you could follow their prostate cancer with active surveillance if it's less aggressive. And then the older you get, then things can be a little bit more aggressive and still not cause very many problems. So we follow things along here. We generally follow the patients along every 6 months with a PSA and a DRE, so just the blood test and a finger, essentially just checking the prostate, looking for nodules or any aggressive disease, and then biopsies intermittently. Certainly we start off more aggressively with biopsies in the first couple of years, and failure would result in increase in Gleason score. So if you remember back to that slide where I showed you before, the dark bar where the more aggressive you have, the more aggressive the prostate cancer is and the more problems it's going to cause. So failure in an active surveillance would be if your Gleason score jumped up to 7, 8 or higher. So and part of this trial that we have, or part of the active surveillance protocols, we're actually doing research, and Morley spoke a little bit about that, and I'll talk more about the metabolomics, but we have this fantastic resource at U of C that funds this tumor banking, and we're able to collect samples on a 6-month basis, and it's completely funded partly by the Alberta Cancer Foundation and other agencies around the province, which is an incredible resource, and certainly we're very lucky to have it here in Calgary and Edmonton, and excuse me, would be, if funded on the cost of this would be in the order of millions of dollars to actually get this done. All right, so the critical question is, when we follow these people along, and I talked to you a little bit about failure, if the biopsy becomes more aggressive, then essentially you need to get into action. You may need surgery, radiation, or other definitive treatment, and about 30% of the population within active surveillance is going to develop more aggressive disease. And it's not clear why this happens, but this is a critical question. So if we can answer that up front, by looking at the cell signaling that was just discussed, or looking at it with the research question, looking at metabolomics, then perhaps we could tell up front, you get an elevated PSA, you get a biopsy, you have a Gleason 6, is there a subset of that population that we know is going to develop onto more aggressive disease, and perhaps we could treat them early? And then maybe there's a 70% of the population that is just going to be fine, and we can leave them alone. We don't need to follow them along every year with a biopsy or every 2 years with a biopsy, because as many of you know, there are side effects and problems with biopsies. So that's the critical question that we're all looking at, and I think the active surveillance is a very good population to answer that. All right, so now we've talked about metabolomics a little bit, and I'll try and explain what it is. It's part of systems biology. So if you think back, or you don't have to think back, I guess your parents were responsible for creating your DNA, and this is the mapping for everything. It's also the mapping and coding for enzymes that Morley has talked a little bit about. So the DNA gets coded into this other molecule called mRNA or RNA, which then gets coded into proteins, and then the proteins can direct metabolites. So it's something like glucose or many amino acids and things like that. And there's thousands of metabolites. There we go. That this picture essentially shows. So it's all these thousands of tiny little molecules that are regulated in part by enzymes and different cascades. So metabolism or metabolomics looks at a whole bunch of these different enzymes, these different components. And if we're able to have a population of benign disease, so a population with, say, BPH, for example, or low-grade cancer, and we're able to compare them to a population with high-grade cancer, and we're able to look at hundreds of these different molecules, then some are going to be up and some are going to be down. And the cancer cells we know are going to grow faster. So it's like essentially an engine with the accelerator on, full speed. And if we looked at the exhaust, which would be similar to looking at hundreds of these different little molecules, then we might find a pattern. So you can imagine with BPH, you might get little spikes, and all these spikes would represent different small molecules. These are specialized MRIs, essentially. It would be the same MRI that we actually look at to diagnose kidney cancer or something like that, or look at your joints. However, they're much more sensitive and much more powerful. To put these things in perspective, Hans was telling me about a floor down, there was a maintenance person at the U of C who was reaching up with all his metal equipment to repair the fire system. And as he got up closer to the magnet, which was through a cement floor above him, all of his tools started to shake and then eventually stuck to the roof, and he went running out of the building, thought it was possessed. So they're very, very powerful, very expensive tools, and we're lucky to have Hans. So what they can do is you get a biosample here. It doesn't really matter what it is. Very, very strong magnet, and you can appreciate how strong that is. I'm sure that workman can. That aligns all of these molecules up. So all of the different molecules or atoms have a specific charge. If you put them in a really small and really strong magnet, they line up on one pole. Okay, so they're all just lined up as if you were to put a magnet essentially on iron filing, so they all just point one direction. So then a signal, a radio frequency signal, comes in and knocks them off this alignment. And as they come back, they emit this signal, and we're able to detect that signal, and then you can tell what these different compounds are. Okay, so it's very similar to MRI, and you get a spectrum like this, and once you know what these different compounds are, you get valine, ethanol, lactate, alanine, arginine, all these different things, and the different height of the peaks are going to be how much concentration is in there. And this goes on for hundreds of molecules. We'll probably leave it at that there. So the principle is that these malignant cells are going to be different than the benign cells that I discussed before. And we can do this in serum, urine, post-diureses, and semen and other tissue. So that's part of the goal. I'll probably move on from that. The benefit of the MRI or NMR, which is similar to the NMR, is it's very simple. So you can collect the blood, separate it, filter it down, and then just throw it into the machine. And that's a fairly important detail. I think that's going to be a useful tool, and we're working towards that. There's one other area that this metabolomics might be very useful, and this is a study that looked at metastases. So the important thing, and cancer causes problems if it moves out of the prostate into other areas. And one of the things that prostate cancer, one of the places it loves to go, is the bones. And this study looked at metabolomics in bony metastases, and they were able to differentiate in serum the differences between people who had bony metastases and those who didn't. So generally today we do that with a bone scan, but it's not very sensitive. Bone scans, while useful if you've got a whole bunch of cells involved in the bones or a whole bunch of metastases in the bones, it's not all that sensitive. So if we're able to increase the sensitivity with metabolomics through this, that would have huge clinical implications, and we might not need to do surgery on people who aren't going to benefit it, or we can start treatment early with metastases. So I think this is just a very promising area where metabolomics might be somewhat useful. So Ulumi Falagan is a PhD student who's working in hands, and she's looked at adding different compounds. So different compounds that you saw in that very complex graph. One of them is alanine, glycine, and sarcosine. So these are essentially different amino acids. And we were looking at how they affect growth of prostate cancer cells. So here we have different days. I think it was day 4, 8, 16, and 24. When you plate cells, and these happen to be prostate cancer cells in a media, so this is the 3D gel, they're going to grow normally and with time. And I want you to, if you can all see, this is generally a round cell. So this is not a very aggressive cell. We didn't add anything special into here, and the prostate cancer cells just grew around. So this would be a normal prostate cancer cell in your prostate that isn't spreading or isn't metastasizing. Alanine is another amino acid, so this is another control. And as time goes along, the cancer cells grow, and they're still fairly normal at about day 24. But if you look at glycine, which is a compound that's upstream to sarcosine, you can see, and I hope you can appreciate the difference. With time, the cells grow, but if you look at this cell, it's got all these little pieces hanging out here. Okay, so it's like if you've seen videos of amoebas where they just have little pieces of their cell that are moving. So these are like jello that's moving through the matrix, and this suggests that this prostate cell is more aggressive and more invasive. So another compound, again related to glycine, is called sarcosine. Same thing here. We're stepping along with growth of cells. And if you remember, in the last slide, we only saw these little growths on the 4th component, so day 24. Stepping a little bit earlier, you can see all these little finger tentacles going out, and the cells are moving through the matrix, so they're more likely to metastasize. And that's some of the principles here with, if we can measure all these different compounds with sarcosine, glycine, and then map them all out, then again, we may have a signal or a diagnostic test to determine which cells are going to move out of the prostate and into the bone, and that's when cancer causes problems, when it moves outside the prostate into other areas. So I'm happy to answer any questions that you might have regarding any of those.
Yeah, so I've been doing active surveillance for 4.5 years, and I enrolled in your study earlier this year. So I'm assuming that all those samples that I gave you went into the study are...
Well, yeah, we're looking at analyzing them, yeah.
Okay. But if I read your slide correctly about the protocol for active surveillance, I was 55 when I was diagnosed with Gleason 6. Now it appears to have progressed to Gleason 7, and I'm essentially 60. Does that, and if I read it correctly, I failed the protocol, so there's no point in continuing in the study, or is there any point in going further?
You mean in continuing within, like,
would there be any scientific validity for me to continue, or am I just risking my life for no purpose if I decided not to treat it? continuing active surveillance.
Yeah, it's tough talking about personal things in a whole group. Certainly, as you develop, go from more aggressive disease from Gleason 6 to Gleason 7, then it certainly does become more significant.
But what I mean is there any benefit for that particular case being in the study or failing?
Yeah, I think that's probably a discussion that we'd have to have in the office per se, but people have to decide what they want and which treatment they want, and there's a variety of different treatments for a variety of different Gleason scores. But certainly a younger individual such as yourself with Gleason 7 disease, it might be beneficial to have treatment in the form of surgery, radiation, brachytherapy and seeds, such like that.
Let me just add a quick comment to that. Whatever your decision will be, it's your decision, no question.
Oh, yeah.
No. And you're going to get the best treatment anywhere in the world because we tuned into anywhere in the world. But don't feel that whatever decision you make is going to hamper what we do. Mm-hmm. The issue is that as you make your decision, you will be followed. You decide to go one way or the other. That's between you and your physician.
Yeah.
But all of the data that has been accumulated as you've gone on will become part of the story. So let's suppose a person in your situation said, you know what, I'm going to play the odds, I want surgery tomorrow, fine. You get your biopsies, you go to surgery, or you go to surgery and the tissue gets analyzed, you're still part of the study. Okay. You start, you know what, I don't want to put up with the morbidity of an operation, I'm going to give it another go period. And come back. That decision too, you're still on board. So either way, it's a win-win situation is what I'm trying to say in terms of you contributing to what we're trying to look for. Okay. And you win because you get the chance to make the choice that's right for you. So I don't know if I've said it perfectly.
No, I think that was clarified.
Yeah, and I wasn't sure if you're asking if your contributions to the active surveillance were not helpful. Is that? Or, no, unfortunately, they're very helpful because if you do choose treatment, then we're going to have that information relayed to that. So it definitely wasn't. Yeah. I'm caught in a little bit of a dilemma because I can't.
I understand.
Yeah.
What kind of a timeline do you see as developing a test to demonstrate whether the cancer is aggressive or not? Are you talking 5 years, 10 years, 15 years down the road?
I don't have a solid answer there. I'm always more Yeah. It always takes a little bit longer to develop into clinical practice. So going from the lab or even from looking at these metabolomics, taking it to the next step and validating it, and then bringing it mainstream, can take as long as 10 to 15 years. Certainly there are multiple different other research endeavors that are going on in different avenues, looking at genetic markers for this that may come to fruition hopefully sooner than that. Go ahead, yes.
Sure. Thank you, man. I guess I'm chair of the Warriors Group because we probably have about 40 specimens here that we can present to you as individuals. What I want to challenge you to think about is how you, as an institution and as a network, communicate and relate back to your receptor audience. And I was just reading very recently, this past week, about Mayo Clinic and how it has a center of individualized medicine. And I think a lot of that is smoke and mirrors in some respects. In other words, it's a little bit of marketing and advertising. But what they want to do is to take their research and quickly relate it to things happening in the clinic. and to learn across those two areas, and to bridge across the two. And so they appear to me to be able to take information that they can garner from people in research trials and the likes, and put that back into the researcher and vice versa. I posed that very same question when I visited the BC prostate cancer group, and my sense is that they're endeavoring to get from the research lab at least information and samples back to the receptors, the users, and to learn from that much more quickly than maybe what we are doing in Calgary. And are you constrained by, how are you constrained in accomplishing the same thing in a very public way? Because like I say, we've got 40 to 50 specimens in this room, ready and willing to engage you and to be with you in your mission and you in our mission.
I guess I would disagree in some ways. Having worked down in the States for a number of years, I have some experience in there, and not to knock them. There are some world-class centers. Mayo would be one of them. MSK in New York, Hopkins, certainly, all fantastic centers, and their research is superb. I think there's been a shift recently here in Canada and in Alberta and in Calgary, and a lot of it has come from Prostate Cancer Canada and from Movember and the funding that we have. Canada generates a huge amount of funding from Movember. So this is giving us a huge boost. Also, we are advantaged because this is a public health care system. There's 15 urologists working in Calgary, and we capture 1.5 million people. Okay, if we compare that to the number of urologists who are working, say, in Baltimore, there's about 50 for 500,000 and people. So our capture option in terms of, and I'm talking specifically in research here, is massive compared to those centers. If we were to look at a population of Alberta, 3 million people, and we have a huge group with John Lewis and a lot of those team members looking on there, all coming together. And I think it's happening, then we're going to have a bigger population of research probably than anywhere in the world. So that's a huge advantage that we have. Does that answer your question to some degree?
My first point. The point is there's not recognition of that. And my second question would be, this community will raise $10 million in favor this year, in this month, for Movember, out of maybe 50 million in Canada. Do you have a corresponding ambition to use productively $10 million in research, and do you have that capacity?
Yes, is one of the grants that we're putting in is for 11 million. That's not directly related to Movember. So what that does, and I agree with them to some respects, is all that money goes back, and then they have a funding agency that's reviewed by a bunch of people, and that's spun out. So that has to happen to some degree. So absolutely, we can use $10 million, no problem.
40 specimens here ready.
Yeah, and you're welcome to come up to and donate at any time. I need more people. That's at the Prostate Cancer Center. Daleen will sign you up.
We don't have the sense that there is that corresponding effort relative to dollars.
Yeah. Well, I guess we need your help. We need to communicate better to you, and perhaps you can communicate better to us as well.
I'd like to compliment a little bit what Eric's saying. Do you know that some of the issues you raised?
Quiet, please.
So the question is, are we doing as well as, worse than, etc., than the Mayo? I can answer that. We're doing just about as well as they do in a lot of areas, and I'll speak from my father's experience. My dad was a physician surgeon in Winnipeg. He was a superb diagnostician, and he had the wisdom that when he made a good diagnosis, he would refer them to the Mayo, so that the Mayo would tell the patient that my dad was right. And that happened a lot. So what I'd like to emphasize is that because of Lockheed's investment in the Heritage Medical Research Foundation, the venue here is as good as anywhere. And the advantage is that people like Eric and myself have been there. We've seen what goes on at the Hopkins and the Mayos of this world. We know what to expect. And everybody here has as much drive as people at Hopkins and the Mayo to bring the discovery that we might make in the laboratory as rapidly as possible to benefit the patient. There is no inhibition, and I'm here because of the lack of resistance to bringing Eric and I are communicating as physician, surgeon, basic scientists, nothing in our way except the hours of the day. The question was, so how long is it going to take? And that's on everybody's mind. And I'll say, you know what? It's as difficult to predict, and I don't like to put this in a negative way, as a physician tells as asked by the patient, so doc, I've got colon cancer, how long am I going to live? I don't think that any physician can give an appropriate answer because basically they do not know. It's a matter, in a sense, of serendipity, wisdom, and chance. If she decides tomorrow that our laboratory is going to come on something that nobody thought of, we could bring that to you pretty quickly, and we would bring it faster in Calgary than elsewhere. But if something became available elsewhere, I hate to put it in a negative way, it would be here like a dirty shirt tomorrow, just like the latest drug on the street that turns up in Los Angeles is here in Calgary tomorrow. So the communication is very good. I'm working with people, I've never met this guy Lewis. I met him on the internet, and not in a negative way, okay? So the deal is that what people in our group, and I consider myself part of your group, have to know is that the communication is wide open, and there's no barrier to progress except the lack of knowledge. So, can we use 10 million a year to further the process? The answer is yes, but I don't want the 10 million, to be honest, unless I know that I'm going to get the return on the dollar that you all deserve. And to me, the biggest impediment that we have in our institution is the lack of stability, meaning that a lot of people at the faculty and many people in the United States are depending on the research results to pay their rent. Not quite in the faculty, but they're trying to balance out how many people can we keep on, and I don't want to get into the politics of the Heritage Foundation shooting itself in the brains out, but the issue is that stability is important. So if you raise 10 million this year, I'm not going to use it this year. I want to put it into capacity so that we get a bright student like Remy come along. We don't have to hesitate, hem and haw, and say, well, I don't get my grant next year, then I'm out of business. So I think that the answer to the money raising thing is to have some kind of capacitor in the system. That means that when the plug goes out, you've got a battery in the radio that keeps it playing. That's what a capacitor is. It stores energy to be used at times when you don't have energy coming in from outside. So the money is essential. If I was not fortunate enough to get the Movember grant, I would have had to start thinking about hiring a techie who's been with me for 15 years. So that's part of the process. But your comment about communication is really important. I'm here tonight because it is so essential that you become part of the solution to the problem. I don't want the 40 samples today because I don't know what the hell we're going to do with them today. But I sure want them tomorrow when we figure out what's going on in this conference. So that's why I think you, the audience, and we working at the bench, make part of the team. And if you become part of the team, You know, you can say, hey, Hollenberg, you know, what'd you do with that $100,000 last year? So I used to tell my father, I said, well, you know, I solved the problem of colitis and arthritis, which are a mouse. Now we've got to work on people. So the communication is absolutely essential. And I think it's not bad, actually.
Yeah, it certainly can be. The other thing I mean, we're not really competing with those institutions as well. You know, having been down there, we're still collaborating and working with them and using their strengths, and they're in part using our strengths as well. So it's not just Canada, the U.S. anymore. You know, it's from Europe as well.
You're as close to Discovery Hopkins by living in Calgary as Hopkins is to Calgary living in Vaughan.
Well, we know that as warriors. I guess the point I'm leaving you to think about is, as an institution, how are you conveying yourself to the public? And are you conveying your message in the very same way and better way than the John Hopkins or the Harvard or the Mayo Clinic? And they have in Mayo Clinic this so-called Center of Personalized Medicine. That means something to people with advanced prostate cancer. It drives them. It's meaningful. It's also meaningful in terms of money, even though the solution may not be for them. Just how you present yourself to the community is very different than how those institutions present themselves to the community. And part of that is because they're out hustling money through their...
It's a huge part of it, advertisement, yes.
But why is that a barrier for you?
I don't think it's a barrier. I think that
Don't let it be a
you just
barrier.
find yourself a job.
Yeah, don't let it be
I agree. You're right.
a barrier.
No, it's not a barrier. Let me tell you what it's about. It's about not knowing how to do it. Okay?
Okay.
I have to share this with you. When I came from Baltimore to Calgary, what I realized is that if I look at the way Hopkins goes and raises money, and if I look at what's going on across the campus, they hadn't a clue, no idea how to do it. That's what you're facing. It's not that they don't want to do it, it's that they don't know how to do it. And I think we need coaching. I think the issue of individualized medicine is a lot of smoke and mirrors, as you say. But I think that the university in general and our faculty specifically could do a lot better about communicating what we're doing on the outside. And they're making it. So there's a downtown, I learned that there's a downtown course that I can go and teach. I thought of it, so how's this one? It's called Sex, Drugs, and Alcohol. I think you might come and hear about pharmacokinetics, pharmacodynamics, opiate addiction, and all that kind of thing. So there are efforts on that behalf, but it's not done well enough to make you feel that you have bought into something that's going through. I'm here because that's the message I want to give you. We got a good show going, and having been in Calgary since '79, you've got a good deal going in Calgary, and it's because of the Heritage Foundation for Medical Research. They started it, they put the seeds in the ground, they watered the ground, and I don't want to get into the mistakes they're making today, but we have a good deal going in Alberta for sure. That's why I'm here. And I don't think we communicated well enough on the outside. And we need some coaching.
And yeah, we're happy to take it or any advice or help, yeah, for sure.
Question?
I had a question. I never got a chance to ask the question.
Quick
It's very
question.
simple.
Go for it.
Very simple. Why do we not hear enough, or I should say more, about what you call the HIFU procedures?
Yeah.
I mean, that's a very, very good method. I personally think that, because I have it myself, and it's now it's 8 years ago, and I'm fine. why do we not hear more about it? How's the research coming along on HIFU?
Well, there's a number of centers that are doing HIFU. Toronto does HIFU, it's a fee for service. It's not FDA approved in the U.S. For a number of reasons. They haven't thought it's come to the table with surgery or with radiation.
But they have about 10 different locations in the U.S.
Yep.
or more maybe. It's about 7 years ago since I did my research on it.
Yeah, I'm not sure that the long-term data is there, comparing it to surgery and radiation.
But
That's what I've seen.
we have over 180 centers in the world doing it. So I just don't understand why do we only have one facility in Canada?
Again, it really hasn't reached widespread acceptance in physicians and the scientific
It's
community.
been going on since 1982.
HIFU is a high frequency ultrasound. So what you do is you stick probes into the prostate and then it's able to heat up the prostate and essentially kill the cancer cells.
See, a lot of people here don't even know about it.
I just don't, some of the data in the last EUA meeting hasn't shown that it's as successful as the
So, is plans to do some more research on that?
Not here, no.
I'm going to stop questions now, but I do welcome people to stay afterwards to ask more questions. I would like to address that I think we're lucky to have the research in Canada that we do. I mean, both of our presenters today were trained at Johns Hopkins, which is recognized as a world-class facility in research and medicine, and yet they've chosen to work here in Calgary. And I also think it behooves all of us to spread the word on how good our healthcare system is, despite our governments, and how good the researchers and the clinicians are that we've got here. So before we go any further with the rest of my short slide presentation, I'd just like you all to join me in thanking our two world-class researchers in